Okay, so this is part 3. I'm going to skip ahead in the book a little bit to page 344 and first talk about animal viruses. With animal virus replication and 1, 2, 3, 4, 5 steps. The animal virus sticks to the cell. It gets inside the cell, so attachment. Gets inside the cell, penetration. Many times it hijacks the cell's machines and builds pieces of itself. That's the synthesis part. It has to put those pieces together to construct new virus particles, new virions, and then those new virus particles have to escape the cell so that they can go on and infect other cells nearby. So on to slide 12 here. Looking at the 5-step infection cycle. Number 1, attachment. So we looked a second ago at our virus with its spikes. For example, here, this S protein, the S stands for spike. And it is these spikes that are important. Spikes, in this case, embedded in the envelope of an envelope virus, or spikes, in this case, attached directly to the capsid of a non-envelope virus. And these spikes are proteins. And proteins are essential because of the ability of proteins to make shapes. So go back to the idea of a gene into messenger RNA to a ribosome, decoded into an amino acid sequence, and that amino acid sequence folds up. And a different amino acid sequence will fold up in different ways. So one amino acid sequence will fold up into a triangle, and one amino acid sequence will fold up into a square, in a very simple way of looking at it. So proteins and proteins' ability to match shapes. Shape matching is something we've looked at before in lots of instances, and it's very important in the way that viruses work. Our cells in our body have all kinds of things on them. So on the outer surface of our cells we have our lipid membrane, our lipid bilayer, I won't draw the entire bilayer, but also on the surface of our cells, this is inside the cell, this is outside the cell, stuck into the membrane, sometimes going across the membrane, sometimes just kind of embedded in the membrane. We have proteins. And sometimes those proteins have stuff stuck on them too. And one important kind of structure on a cell surface is a glycoprotein. So let's look at a membrane. Let's look at our protein stuck in that membrane and folded up in its particular shape. And then let's attach a sugar molecule to that protein. And now we have our protein here in the green, we have our sugar molecule in the red, and now we have a glycoprotein stuck in our membrane. And you can see that the protein and the sugar have created this kind of shape, this shape that the virus is going to take advantage of. On the end of our virus's spike, it has a protein that perfectly fits into the gap. In this case, we would call this protein, this glycoprotein, a receptor, and a viral spike fits into that receptor. And there's a lot of variety in the way that a cell can interact, or indeed a virus can interact with a cell. Sometimes, or often, more than one receptor is required. For example, HIV in the picture here requires 2 different molecules for the virus to successfully stick and to get and to stick onto the cells, and the virus has to get stuck, otherwise it can't get in. HIV in particular binds to these things called CD4 cells. If you remember our immune cells, CD4 cells were another name for helper T cells. So proteins with carbohydrates attached act in cell-to-cell recognition and binding of molecules. So one thing to be wary of is that although we would describe this as the receptor for the virus, that's not the job of that glycoprotein. There are no molecules on the surface of your cell that want to attract viruses. It's just that the virus has taken advantage of all these different shapes to recognize 1 or 2 of them specifically and latch on. Different parts of your body, different tissue types have different cell surfaces. So all the different various shapes of proteins and glycoproteins, say on a cell in your lung, are different to, say, the glycoproteins and cell stuff that's around your kidneys or around your the cells of your intestines or around the cells of your bladder. So all the different parts in your body, all the different tissues have different cell surfaces. And the ability of a virus then to attack different parts of the body, or even attack different kinds of animals, is then based upon the specificity of the receptors and the virus spikes. If a virus can attach to a receptor, and if that receptor is shared amongst a variety of different cell tissues, then the virus can attack many different kinds of cell tissues. And that is known as tissue tropism, maybe broad or narrow. This limits then where the virus can attack inside the body. So rabies, for example, has a narrow range. It only really affects neuronal tissue, neurons. You and other animals, particularly humans and other mammals, the more away you are. So we have animals, and then we have mammals, and then you have vertebrates. The closer you are, so a human and a pig are pretty close because we're both animals, we're both vertebrates, we're both mammals. A human and a turtle are much more far apart. Even though we are both animals, and we're both vertebrates, one is a reptile and one is a mammal. And then, say, humans and earthworms are very far apart because the earthworm is, even though it's an animal, it's an invertebrate, and the human is a vertebrate. And the closer or further you get, the more similar or less similar your cells are. The reason why, for example, mice can be used quite effectively for studying disease is because they too are mammals, and generally then what affects a mouse cell is going to affect a human cell. This is a second idea behind what's called host range or host tropism. Again, although more dissimilar, a human and a mouse, their cell surfaces are going to have a few similarities, and if the virus has a spike that attaches to one of those receptors that is the same between you and a mouse, then both a human and a mouse can get the disease. If the virus has a receptor that, say, only a human has, then it can only affect the human. Measles, for example. Humans can't give measles to dogs because the dog doesn't have the right receptor for the measles virus, even though dogs and humans, both being mammals, are very similar. Rabies has a broad host range. It infects many unrelated animals. Horses and humans and all kinds of things can suffer from rabies because it targets a receptor that is common to all those different kinds of animals. What about COVID-19? What about SARS-CoV-2? The virus attaches to receptors called ACE2, and it is a transmembrane enzyme. It's one of those proteins. In this case, it's an enzyme that goes all the way across the cell membrane. There's parts of the ACE2 structure outside the cell, and there's parts of the ACE2 structure inside the cell. ACE2 is found on cells in the lungs, which is quite should be no surprise because that is where we see all the symptoms occurring. It's also found on arteries, on the heart, on kidneys, and the intestines. And ACE2, again, is not trying to be a place for the virus to stick. It has an entirely different purpose. In fact, it has quite a wide variety of things that ACE2 does. So here's the coronavirus, here's the cell, here is the transmembrane ACE2, and here are the spike proteins sticking into the ACE2 receptor. One way we could maybe counteract the SARS virus, and viruses in particular, is here is our ACE enzyme. So here's our membrane, it's inside the cell, it's outside the cell. This is our ACE2 protein, and our virus has a spike protein that fits nicely into the ACE2 and allows the virus to stick. If we can build an antibody that sticks to ACE2, and we can attach that antibody onto the ACE2 protein, that then blocks access of the viral spike. And if the virus can't attach, it can't infect. So antibodies used to block the receptor are one way that we can try and fight viruses. Okay, slide 15. Got a little video to show you about the entry of viruses. Hopefully you should be able to hear this at the same time I do. So here we go.
There are two mechanisms by which enveloped viruses enter host cells. In one of the mechanisms, the virion attaches to host cell receptors by specific proteins on its surface called spikes. The envelope of the virus fuses with the plasma membrane of the host, and the nucleocapsid is released directly into the cytoplasm. The nucleic acid then separates from the protein coat. In the second mechanism, the enveloped virus adsorbs to the host cell by specific proteins on its surface, and the virion is taken in by endocytosis. In this process, the host cell plasma membrane surrounds the whole virion and forms a vesicle. The envelope of the virion then fuses with the plasma membrane of the vesicle, and the nucleocapsid is released into the host cytoplasm. The capsid protein is then removed, releasing the nucleic acid of the virus. A naked virion also enters by endocytosis. Since the virus has no envelope, it cannot fuse with the plasma membrane. After being engulfed, the viral nucleic acid is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
Okay, so on to slide 16. Step 2, penetration and uncoating. And this is going to depend on whether our virus has an envelope or if it is naked. If our virus has an envelope, it'll go through that process of fusion. The virus's envelope fuses with the cell envelope, and the virus is taken up. The nucleocapsid is taken up into the cell. One thing to note is that the virus with its envelope, these little brown dots here, these are the viral spikes, and these blue things here are the host receptors. These viral spikes were put into the membrane as part of the assembly and the release of the virus, which we'll look at in a second. But you'll see that as the virus enters and as the envelope, the membrane of the virus, fuses with the membrane of the cell, you'll see it leaves behind telltale signs. It leaves behind these spikes in the host membrane. And this is an important way that our immune system can recognize a virus-infected cell. Our immune system is looking at the surface of cells, checking them out for anything suspicious. And these viral spikes left behind as a virus penetrates are a giveaway sign. We can send antibodies against these spike proteins, or maybe they can be recognized by other cells in the system, such as cytotoxic T cells. If you are enveloped, you have a second method of being uptaken called endocytosis. So endo meaning inside, cyte meaning vessel, osis meaning the process. So the process of taking things in using a vessel. So we have our attachment, our spike proteins sticking to receptors, but instead of the membranes fusing, the cell engulfs the viral particle and pulls the entire thing inside and puts it in a vesicle. And then later on, the nucleocapsid escapes that vesicle and releases its nucleic acids. This is how non-envelope viruses rely upon a cell getting them in. Our non-envelope viruses stick, their spikes attached directly to the capsid, stick to receptors, and then they are engulfed by the cell and taken inside. Uncoating of the uncoating of the virus happens inside the cell. It may happen in different locations based upon where the virus wants to go.